Let's start off with exactly what we'll cover in this video. We'll begin by understanding what stem cells are and how they differ from specialized cells. Next, we'll examine the different types of stem cells and where they're found. Then we'll explore the medical applications of stem cells and their potential to treat various diseases. Finally, we'll consider some of the ethical debates surrounding stem cell research. So, what exactly are stem cells? To understand this, we need to remember what cell differentiation is. Cell differentiation is the process where a cell becomes specialized for a specific job. For example, some cells differentiate to become muscle cells, while others become nerve cells. Once a cell has differentiated, it generally can't change into a different type of cell. Stem cells, however, are different. They're undifferentiated cells. They have two special abilities that make them unique. First, they can divide to produce more undifferentiated stem cells. Second, they can differentiate into specific specialized cells when given the right signals. This makes them incredibly valuable for medical research and treatments. There are different types of stem cells that can be found in different places. Embryonic stem cells are found in early human embryos, just a few days after fertilization. These cells have the potential to develop into almost any type of cell in the human body. Think about it. Every cell in your body, from brain cells to skin cells, originally came from these early embryonic cells. Adults can also have stem cells, but they're more limited. Adult stem cells can be found in specific areas like bone marrow. Unlike embryonic stem cells, adult stem cells can only differentiate into specific types of cells. For example, bone marrow stem cells can become different types of blood cells, but not nerve cells. Both embryonic and adult stem cells can be grown in laboratories. Scientists can clone them to make genetically identical copies and guide them to differentiate into specific cell types. Stem cells have enormous potential for treating disease and injuries. Adult stem cells are already being used in medicine today. For example, bone marrow transplants use stem cells to replace faulty blood cells in patients. Embryonic stem cells could also potentially be used to replace damaged or dysfunctional cells in many conditions. This could create new insulin producing cells for people with diabetes, or they might generate new nerve cells for people with spinal cord injuries. To make stem cells treatments even more effective, scientists use a technique called therapeutic cloning. In this process, an embryo is created with the same genetic information as the patient. The stem cells from this embryo would be genetically identical to the patient. This means that the patient's immune system wouldn't reject them if they were transplanted. However, there are risks associated with stem cell treatments. For example, stem cells grown in labs might be contaminated with viruses. Stem cell research, particularly embryonic stem cell research, has sparked significant ethical debate. Some people object to embryonic stem cell research on moral grounds. They believe that human embryos deserve protection as potential human lives. They argue that destroying embryos for research violates the sanctity of human life. Others take a different view, focusing on the potential benefits to patients. They point out that embryos used in research are typically extra, unused embryos from fertility clinics. These embryos would likely be discarded if not used for research. They argue that helping existing patients who are suffering should take priority. It's also important to keep in mind that different countries have different regulations regarding stem cell research. In the UK, embryonic stem cell research is permitted but strictly regulated. In some other countries, it's completely banned. Plants also have stem cells, located in regions called meristems, which are located at the tips of the roots and shoots of a plant. Meristem cells remain undifferentiated throughout the plant's entire life and can divide and differentiate into any type of plant cell. This ability makes plant stem cells incredibly useful in agriculture and conservation. They can be used to produce identical copies of plants quickly and economically. This technique can help preserve plants that are resistant to certain diseases, or plants facing the threat of extinction can be cloned. As you can see, stem cell research represents both remarkable potential and significant challenges. Stick around and watch our next episode, which will focus on diffusion and exchange surfaces.